Hello, everyone, and welcome back. Our next speaker of the day, as you could appreciate from the banner previously, is Petar Šćepanović. I hope I'm pronouncing it correctly. And he is a senior scientist at Roche. And Petar today is going to talk about the microbiome in big pharma. So again, an interesting topic, and actually between pharma and a lot of research done here at Nestle. So very interesting topic. And without any further hesitation, I would like to invite Petar onto the stage. Hi, Petar.
Hello.
How are you?
I'm good, thank you. It's a pleasure to be here and to listen to all the talks, and just now in the speaker session. It was really fun.
Was it? Was it good? I'm glad to hear. Yeah. So we're very excited for your presentation, and I see you have some slides ready. Indeed.
Yes.
So
you can see everything. It's working, right?
Yeah, everything. We can hear you. The slides are big on the screen. So I'm looking forward to your presentation, and I give the floor to you.
Perfect. Thank you. So it's really a pleasure to be here, as I just said, to tell you a little bit more about my work. I'll start off with 3 very boring slides about myself, about my path, and then talk a little bit about microbiome in general, and then what are we doing in Roche. So I by the way, I don't have anything on Roche. Marco before talked a little bit more about it. It's a big company. I think you all know it. There are different organizations within Genentech in South San Francisco. I'm based in Basel in a part called PRED, Pharma Early Research and Early Development. So I'll tell you how I got here where I am right now. And this is my career path slide. I'm from Montenegro. I was born and raised there. And I put a map of Europe to point where it is, because it's a small country. So it's fine if people don't know where it is. And especially it's this small little dot. And I realized when I put this image here that I would have to increase it so much that it would go all over the slide to make it visible where it is. But it's right here across Italy, close to Serbia, Croatia. And then after finishing high school there, I went to University of Turin in Italy, where I did my bachelor's and master's in biotechnology and molecular biotechnology. And following that, so I'll go quickly through this. And it really seems like a simple path, but the truth is that there's a lot of failures in there, a lot of different things that were supposed to happen, but didn't end up being like this. So after Torino, I went to Lausanne, did my PhD at EPFL, and then I was a postdoc or a research associate, how they call it, at the University of Cambridge for a year. And then since just a little bit before pandemics, I started working in Roche in Basel as a senior scientist. So it's a little bit more of explanation on how Montenegrin dude ended up in Torino. Seems a little bit random. But I have a twin brother, and he's an aerospace engineer. So we wanted to be in the same city. And when we decided what we wanted to study, there was not tremendously a lot of options, in the sense that we knew that we could have a scholarship in Italy. And Torino was one of the few cities in Italy that had these 2 faculties in the same city. So that's how. And then I had a really amazing time. And I put a similar photo to Marco with Molentonelliana and the Alps in the background. And I spent 5 amazing years in there. And then Lausanne, as I said, it happened after finishing my master's. I started looking for PhDs, and there was really I had some success in my during my master's with work I was doing. So I had a lot of offers, and the project that on which I focused during my PhD was the thing that really inspired me the most to come to Switzerland. Then Cambridge here, I put it at King's College, which is fine, it's good, but I prefer Trinity and Sydney, Sussex, to be honest. But there I also had an amazing time, and it was lovely. It was not extensively long, unfortunately, because I had an offer then to come to Switzerland, back to Switzerland, and start working at Roche. And now I'm really enjoying walks along the Rhine. And Basel is a city which I didn't give a lot of credit before coming, but it's a very pleasant place. So what did I do in all of these places where I've been to? Here's the research experience where I worked. So I started in Torino working in the lab of Carlo Ponzetto, which was a molecular and cell biology lab, and we were focusing on cancer biology. So it was studying pediatric sarcoma called rhabdomyosarcoma. And at the same time, I started developing I wanted to learn more about bioinformatic methods. So I proposed to Carola to do a project where I would develop my skills in bioinformatics, and she loved it. And she sent me to Paulo Proero, who was my professor of bioinformatics. So I started working on a project there, had a little bit of success, as I said, and that was my first venture into the computational biology. And then for my PhD, I was at EPFL, where I was with Professor Jacques Fillet in the Human Genomics of Infection Immunity Laboratory, where it was purely computational work, so genomics, and I will tell you a bit more what I focused on. And then in Cambridge as well, I focused on that path, Human Genomics and Systems Genomics Laboratory with Mike Inoue. And now in Basel, I'm in Bioinformatics and Exploratory Data Analysis Group. So here I put really the people that I interacted with, and I'm not on any single of these photos because they were all taken either before I joined those labs or after or when I left them. But really, I want to point here the luck that I had to be surrounded with all these amazing people, really, from Italy to Lausanne, Cambridge, and now in Basel. And as you can notice now, the group where I'm embedded is by far the most numerous one. We're like almost 40 computational biologists. And I will tell you a bit more later how does the work look like of the group and what I'm focusing on. So here I wanted to list the publications. Once again, 3 boring slides, as I said, because they really can. I can by talking of the publications that I participated to, that I contributed to, I can explain more about my career path. So it's during my time in Italy, I contributed to this study that was published in Life in 2016, and it was really I had a small little story that I could present for my master's when I was applying for PhD positions, and it really gave me an opportunity to then be able to choose between place where I wanted to do my PhD in. And then in Lausanne, among other works that I contributed to, I just pointed the 2 most important ones for me that I led, which were the study on the association of human genetic variants and the response to common pathogen in vaccines, which we published in Genome Medicine. And then what really defined what I'm doing right now is the study on the non-genetic and genetic effects on the gut microbiome in healthy individuals. So, as I said, quite a lot of explanation about my career path can be given through these. As I explained, this was really helpful to present the work that I was doing and then get offers. And then for my postdoc, it actually went through a tweet because I saw Mike tweeted my paper, and then I got in my paper from Genome Medicine, and I got in touch with Mike, and he offered me a position for a postdoc in Cambridge. So that's how that went. And then for my current position, well, it was this paper that we published, and that was really on microbiome that qualified me to be a good candidate for the position that they were looking for in Roche. And then yes, in Cambridge as well, contributed to work on polygenic risk score, something that I'm trying to develop now, and I will tell you a bit more about that as well. So yeah, I want to switch to microbiome and give you a little bit of introduction to that. And here I just have definitions of typical terms that you encounter. So I'll start with microbiota, ecological communities of commensal symbiotic and pathogenic microorganisms, while microbiome represents the ensemble of microbial genomes and products at a given site. So here I always say there's a lot of confusion And these two terms are used interchangeably, and I really want to say is that don't get hung up on that. Who cares? I mean, if you say microbiota or microbiome, we will kind of understand what you're talking about. So this is just two definitions. That's it. Now to the role of the microbiome. What we really have clear evidence is that it's used, that is important for conversion of food into energy, synthesis of essential vitamins, stimulation of the immune system. But if you have been in the field of science in a couple of last years, then you will know that it has been really implicated into everything you can think of. Up to the point that even choosing of your life partner is dependent on your microbiome composition. I will not get into that, but people have looked into those things and gave microbiome some role into various different things. And then two definitions that I always want to point out are 16S rRNA gene sequencing and shotgun metagenomics, because these are the ways you explore microbiome. So 16S RNA gene sequencing is the amplification and sequencing of the variable regions in the 16S ribosomal RNA genes for taxonomic profiling of bacteria in a sample. While shotgun metagenomics is untargeted, that's why it's shotgun sequencing of all beta microbial genomes, that's genomics, that are present in the sample. Okay, so those are the definitions. And now just the characteristics of the gut microbiome in particular, because that's something we're looking into the most, and the field in general is putting the most attention to the gut microbiome. And on average, you can identify around 160 species per person. And this is the data coming from the MetaHit consortium study that was one of the first ones to be set up to look into these things. And then the dominant phyla that you can observe are Bacteroidetes, Firmicutes, Actinobacteria, and Protobacteria. And then in difference to the genomics or genetics that are very similar between all of us, if you look at it overall, microbiome can be very different from one individual to another, up to, you can see differences from 80 to 90%. Even from the niches of your own body, such as gut, as I mentioned, as I'm talking about here, can be very different through your skin microbiome or your oral microbiome. It changes out through lifetime, so it develops, and then it's affected by diet, age, sex, medication, genetics. And yeah, genetics don't, what my work and that of many other groups show, that it's not the strongest influence. What is presumed to be the biggest impact on it is the diet. So that wraps up the characteristics. And then another thing that was very interesting in the field was initially people started guessing that there's many more bacterial cells on the human body than cells of the host itself. That got revised, so the number of cells, as you can see here on the left-hand side, is 1 to 1, more or less. But what definitely did not get revised, and is still the case, is that the number of genes in the microbiome is tremendously bigger than the number of human genes. So effectively, each and every one of us is carrying genetic material that is predominantly made of bacterial genes. So from this study in Cell and Host Microbe in 2019, they identified 45 million non-redundant genes. So on what we use on daily basis in my work is this collection that was published in Nature Biotech a year ago, Unified Human Gastrointestinal Genome Collection, that contains around 200,000 genes. So compared to 20,000 genes that is presumed in a human genome, the numbers here are much bigger. And these are coming for roughly 4.5 thousand species that are present in this collection. And even 70% of those species we cannot culture in the lab. So they don't have the cultured representatives. They encode around 170 million protein sequences, and of those, 40% lack functional mutations. So, I mean, when you hear these numbers, to me at least, this sounds absolutely crazy. And it just points out how little we know about microbiome, and how there's a lot of dark matter out there, and there's a lot of opportunities in the microbiome. And obviously, the potential is huge. When cases like that, you start thinking, what can all that mean? And here, I like this slide that I saw from a review that it was talking about genomics, and talks about translational potential of genomics, but I use it for microbiome because, well, not just because my background is in human genomics, but because it really checks out all the boxes for microbiome as well. So for prognosis, biomarker discovery, huge potential microbiome there, response to treatment, pharmacogenomic markers, stratification, all of these things, drug repurposing, diagnosis, target discovery, risk prediction, and so on. So it really checks out all the boxes here, and that's why the potential is huge. Obviously, a lot of the people understood that, and there's a lot of efforts, as I said, ongoing in the field of microbiome. And now I get to work in Roche, and I want to tell you a little bit more about that. So obviously, we're looking into these things as well. And the first thing to do is to build an infrastructure and capacity in order to investigate microbiome in clinical trials. And that's part of my work. I'm heavily involved into these sort of things where we're trying to generate interest within clinical teams, and then work on all of these different aspects of a clinical trial where you develop protocol language that would involve microbiome sampling and investigation, standard operating procedures for stool, then logistics and processing. You identify CRO that can do sequencing, and then you finally get to the part of the work that I'm mostly interested in, is developing computational analysis, pipelines and analysis that will generate results that can then feed again into generating more interest from clinical trial teams that are working on specific diseases. And this is how it works, as written here, predominantly driven by diet. You take a sample, you ship it off to a site, to a hospital, then gets to Roche, and it's within a clinical trial, then it gets sequenced and then analyzed. The data is analyzed. And here I also wanted to point one important thing, because this is all very novel, and we're looking to making this data from the beginning fair. So findable, accessible, interoperable, reproducible. So we do not want to fix rusty pipes, because that's difficult work, but we really want to start producing this data natively fair. So we're giving a lot of attention to that as well. So here, I just want to separate this part of my work that is really focused on clinical trials. And here, obviously, I'm involved in many of these things, but I have no idea how these. I mean, I'm looking at them now, but I'm not an expert on SOPs or stuff like that. So I'm working in really big teams, and there's a lot of different people who are working on this, and we work together in order to make it happen. So yeah, work within the clinical trial really means working in a big team where you focused on one particular thing. In my case here, microbiome. Someone comes in from procurement, someone comes in from operations, taking care of logistics and so on, CROs and so on. And really, it's the team's effort that makes all of this capable. So that really takes, in my work, a significant part of my time. And then this being Roche is what it is, meaning a very big company with a lot of resources, it really gives you a possibility to do more innovation side of things, more academic-like projects, I would call them, in the sense that we have opportunity to have postdocs and interns and so on. So what we started building is a microbiome team that would work on different, as I mentioned, academic-like profiles. So we're going to have a postdoctoral fellow that we hired through RPF, Roche Postdoctoral Fellowship Program, that will join the group this year. And here we set up the project so they would be sort of disease agnostics, in the sense that we want to apply them, and I'll tell you more about them, to all the disease areas that Roche has interest in. So neuroscience, then oncology, immunology, and so on. And here we want to use the whole genome sequences to study and derive bacterial signatures out of them. So the idea there stems from this paper from Nature that was published a year ago, and from Rob Knight's group in University of San Diego, where they looked into whole genome sequences from oncology patients, and they managed to derive bacterial signatures that they can use for diagnostic approaches. And the goal there is written, ours is to identify these predictive or diagnostic markers that we can use for patient stratifications. And that helps us to build a rationale to include microbiome analysis in future clinical trials across different disease therapeutic areas that Roche has interest in. So that's one example. And then the other example is this one, where we're integrating microbiome and genetics data. So we're going to have an intern from Roche Advanced Analytics Network program that will join the group in 2021, very soon as well. And the premise of this project is that microbiome acts independently of host genetics. So we can use host genetics on one side and microbiome on the other side that really captures the environmental influences in the best possible quantitative way we can measure. And we can put the two together and we can use them to derive a better risk prediction metric. So here we want to, for the host genetic side, we want to use the polygenic score catalog that was developed at the University of Cambridge, at EBI as well, to implement the use of polygenic risk scores. And then on the microbiome side, we wanted to derive something that we call microbiome dysbiosis index, which has already been attempted at being developed. And we put the two together, as I mentioned, to have a better risk prediction metric. And another example of these innovation sort of projects that we have ongoing is the integration of metagenomics data and metabolomics. And here we have a PhD student that is going to join Professors Fiona Powery lab in University of Oxford, and is going to spend a significant amount of time of this PhD project in Basel with us. And the idea is that you can start from metagenomics data, and you can simulate and do in silico modeling starting from that type of data and obtain metabolite concentrations. So metabolites produced by the microbiome are associated with several diseases, and this can give us opportunity to do patient-specific predictions and more importantly, generate hypotheses that can then be mechanistically investigated further down the line. And that's exactly this type of collaboration that we have set up with Fiona, where the student in Basel will be doing this with us computationally, and then in Oxford go into mice models and culture techniques to use in order to test the hypothesis that we derive. So I believe I wrap it up with that. This is the logo of Roche, of the template that we usually use. And I just want to say that Roche is a very nice place because it has a lot of opportunities. It's a big company that really gives accessible. It's way to push forward projects. It's possible. It's not really constrained by that many factors. So it's a nice place where you can do science. And then I hope I managed to explain to you the side of clinical trials, how they run. And from the group where I'm working now, a lot of them are supporting clinical projects, clinical trials. And then you have all of this other side of innovation that you can have with interns and postdocs and so on that can really bring help you do cool science. So that's it. Thank you all very much.
Thank you, Petar. Very, very interesting presentation, and then nice to see again what computational work is like in the industry setting. I think that's again a new, fresh type of research that we haven't seen yet today. There are a few questions from the audience, and one of the question is, how much bioinformatic tools are developed in-house at Roche? So how long until pipelines or programs are bought or licensed as other software products?
Mm. So we do not tend to. Depends. What we put a lot of emphasis is automation. So if it's really a pipeline that goes from raw reads to getting the counts, having scripts or packages that can automate things, it's extremely useful. So that is developed heavily. So it could be accessible to a lot of different clinical scientists, for example. About developing tools of our own, we tend to use what is in the public domain. So we're not going to start developing Chime, for example, that is used in microbiome data, or Bowtie in host genetics side of things. So we will really use those. Regarding then, for example, licenses and so on, as I said, the resources are usually there, and we do have access to, for example, Keck Pathway database. For which academic doesn't need the license, we bought it and we can use it. So all of those things, it's very comparable to an academic setting, I would say.
Okay, interesting. Thank you very much for the answer, and thank you very much again for your presentation. Do you have any final comments for the audience?
No, if there's any questions left, feel free to write to me. I didn't leave my email there, but it's just my name dot my last name at raj.com. So feel free to send me an email with anything that you might be interested in.
Excellent. It's well received. And also, again, for the participants and also for you, Petar, at 5 o'clock, we will be having another one of our sessions. Hopefully this time less technical issues. And if you would have time to join, Petar, that would be fantastic. And also for the participants, then your remaining questions can be asked in this session. So big thanks again, Petar.
Thank you.
And see you later, hopefully. Bye-bye. All right, so moving along in the schedule, we will move swiftly forward to our next speaker, and see you in a bit.